Hello, everybody. On behalf of the entire Xconomy staff, welcome to Xconomy's 2020 National Awards Program. Now, normally we'd be gathering in a packed ballroom to honor the very best our industry has to offer, to celebrate our most impactful accomplishments. But as we all know, these are not normal times. The safety and well-being of our friends and colleagues is paramount. So instead of coming together in person, Xconomy decided to create a virtual awards festival, starting with this Meet the Finalists webcast series that allows the industry to get to know the 40 companies and individuals that represent the finalists of the 2020 Xconomy National Awards. Over the course of the days leading up to the awards ceremony on September 24th, Xconomy invites you to meet the 2020 finalists for each award category, to hear their stories, to understand their unique game-changing contributions, and recognize the impact they've made to improve our ability to discover, develop, and deliver effective therapeutics and vaccines to those most in need. This year, Xconomy received a record 600 nominations, representing 10 award categories. Each nomination was carefully reviewed and ranked by Xconomy's editorial staff using specific criteria to yield the 4 finalists in each category. But before we get started, please keep in mind that everyone has been registered and provided access to the awards networking app. It's open right now and will remain open through September 24th. It's a great, easy-to-use program that provides all attendees with the opportunity to meet and greet and interact with the other attendees, finalists, sponsors, friends, colleagues, existing and future business partners. Today, we're happy to introduce you to the Big Idea finalists. Revolutions begin with a bold, unique idea. It's the scientific observation that spawns a new way of making drugs, the creative new business strategy that alters the healthcare system, and the technological innovation that promises to improve patient outcomes. The Big Idea Award honors the people, the companies, and organizations who are advancing transformative ideas. The finalists for the 2020 Big Idea Award are Geisse, Curi Bio, Katy, Rezvani, the University of Texas, MD Anderson Cancer Center, Seer, and SQZ Biotech. So let's go ahead and meet our finalists right now.
Thank you so much for your consideration. It's really an honor for us to be grouped in with these other fantastic nominees. So the problem that we're addressing at Curi Bio is that cells in the dish don't look like cells in the body. What I mean by that is that the tools we use in laboratory research don't accurately model human function. And that makes it difficult to find new cures for diseases. When you make therapies, you really need to model how it's going to work in the body before you actually put it into a person. This is really important for like drug development industry, for example. It takes anywhere from 10 to 12 years and $3 billion to get a drug to market. And typically you start out with tens of thousands, hundreds of thousands, million compounds and whittle it down to one drug that gets to market. At the beginning, you use a lot of single molecule assays at high throughput. Towards the end, you use more complex but lower throughput assays. What we're doing is we're using stem cells, which are kind of like a happy medium between those 2 points. Induced pluripotent stem cells can be ethically sourced from blood or urine of a patient. They can be easily scaled up into many human relevant systems that we can use to test a lot of drugs at the same time. Now, while this is a super promising technology, one of the issues that we have is that stem cells, they just don't fully mature into the kinds of tissues we're interested in studying. For example, heart cells, they're very immature in the dish. And that's because, in the womb, there's a lot of stuff that happens that doesn't happen in a petri dish. About 6 weeks after gestation, the tissues that become your heart start beating. And there's all sorts of other signals like hormones, chemicals, structural signals that go into making your heart develop and function properly. So what our team at Curi Bio is doing is we're applying a variety of bioengineering techniques to really mimic these signals in the dish. And the idea there is to really trick the cells into thinking they're back in the body and into making more mature representative tissues. These cells will then enable researchers to test new therapies in ways that would be impossible with single molecule assays or even complex animal models. And they can use what they learn to inform future studies. We're really doing this in a couple of different ways. First, we build model heart tissues to test if a drug causes an arrhythmia, which is like an interference in the regular heart beating. Arrhythmias happen because of a lot of, as a side effect of a lot of drugs, including like allergy drugs, things like that. And a lot of times you really just can't detect that until you put it into a person. And sometimes it can be deadly. It can stop the heart. So we're building cardiac tissues from stem cells. And that's, been tried a lot of times, but in their current state, they're really not able to flag certain drugs that we know to be dangerous. What we've done is we've added bioengineering into something called the Curi assay, which is the cardiac in vitro assay. And that's a platform specifically to test for cardiac problems. It's much better at testing these, flagging these toxicities for these known drugs that typically don't respond to stem cell systems. And we can do this at high throughput. The second thing we're doing is we're starting to model diseases in the dish, particularly muscular dystrophy. You've probably heard the term muscular dystrophy. It's not a single disease. It's a wide classification of a number of childhood diseases, all of which lead to muscle wasting and loss and a very early death. It's a terrible and devastating disease. It's really difficult to model this because muscle contraction is a really complex process. It involves a symphony of biological systems working in harmony to create movement. So developing therapies for this disease really requires a complex model system. Now, animals are great. They take a lot of time, a lot of money to develop, and they can fail to be predictive. 10 years ago, there was a drug that looked super great in a mouse model. Once they gave it to patients, that actually didn't do anything. So here we're using stem cells, but much like before with the heart, the cells alone can't recreate the symphony of muscle movement. So we add our proprietary bioengineering techniques to build complex, biologically relevant models of these patients' muscles and directly test whether a new drug or therapy can restore their contraction. And because we're using induced stem cells, we can actually get this from a single patient. Even if they have a totally unique form of muscular dystrophy, we can model that person in the lab. We call this a concept an N of 1 clinical trial in a dish. Our goal is to use this to speed delivery of new treatments to these patients. And this isn't, what we're doing is important in the industry because we're really doing it at scale. Bioengineering isn't really a new concept. It's been around for quite a bit, but typically it's done to a single data point, build one model system. Employing it in a way where you can test tens of thousands of compounds, hundreds of thousands of compounds is really challenging, and that's what we've really done. So we've scaled these technologies to enable researchers to get better human data earlier in the process. It's going to improve the safety and efficacy of medicine, reduce the number of animals, and really it's going to get these life-saving treatments to market faster for diseases that have patient populations of millions or even patient populations of one. And I'm really proud of my team because as a team, we're really sitting at the crossroads of delivering on the promise that bioengineering has. We've got a fantastic group of stem cell biologists and engineers working together to democratize this technology so that everyone in the field can benefit from bioengineering.
Thank you very much for this opportunity. I've been nominated for work that we performed at MD Anderson Cancer Center, where we established the use of chimeric antigen receptor modified natural killer cells for the treatment of different types of cancer. So just to give you more detail, my group led the development of MD Anderson's current K-cell therapy platform, where we take natural killer cells, which are a type of immune cells from donated umbilical cord blood leftover from birth, and then we genetically engineer these cells to express CARs against specific cancer targets, such as CD19, which is expressed on malignant B cells. In my opinion, this is very important to industry, given that our novel cell-based immunotherapy can provide an impactful off-the-shelf treatment option for patients with certain types of cancer type, and this has not really been offered to patients before. And this can have a major impact on patient care. And it can be seen in a number of ways that I'm going to very kind of briefly summarize. One is that our current K cells are non-related to patient. And what that means is that they are taken from an individual, a healthy donor who is not related to the patient. This makes access to the cells much easier than it was before. And also then extends the approach of immunotherapy or CAR therapy in our case to an off-the-shelf option which could then eliminate some of the hurdles with CAR T-cell therapies that are currently FDA approved, because those are usually patient-specific, and with this, it means that there is a cost of therapy and also the logistics. The second way I think it's impactful is that we've shown that because we've seen minimal toxicities with our CAR NK cells, and by toxicities I mean the toxicities that have been described with CAR T-cell therapy, such as cytokine release syndrome, etc., we can actually administer these cell therapy product, the CAR NK cells, in an outpatient setting, which makes it even more attractive for many patients given the overall reduction in cost of therapy associated with treatment. And thirdly, I've already alluded to the safety profile of our approach. None of our patients to date have experienced any major toxicities such as cytokine release syndrome or neurotoxicity that's been observed with the existing FDA-approved CAR T-cell therapy. And indeed, you know, the potential of our work was recognized recently by Takeda Pharmaceutical, which is one of the largest pharmaceutical companies in Asia, and which entered into an exclusive licensing agreement and research agreement with MD Anderson, really to develop and commercialize our clinical CAR NK cell platform that I mentioned, but also to partner together to develop CAR NK cell therapies for other types of cancer. You know, I think what led really to this nomination was the impressive clinical results that we saw in our phase 1/2 clinical trial that was recently reported in the New England Journal of Medicine, where we showed that our CD19 CAR NK cells in a group of patients, I mean, granted it was a small number of patients, having said that, these were patients with very advanced types of blood cancer, non-Hodgkin lymphoma, and chronic lymphocytic leukemia, where we observed response rates of greater than 75%. And actually 7 of 11 patients achieved complete remission. So, you know, this kind of product, this type of therapy gives new hope to patients who've already failed multiple lines of treatment, as in the case of our clinical trial. And it addresses a current critical unmet need in the field, and that we need safer, cheaper, and more accessible therapies for patients with cancer. And if we were recognized for our work by being awarded this, you know, prestigious award, it further really undermines the type of innovation that is coming out of academic institutions such as ours, and also it will give hope to patients to see that we are working very hard to develop these sorts of therapies for them.
Thrilled and honored to be selected as a finalist in the Big Idea category. Let me tell you a bit about Seer. We're focused on developing transformative products in the field of proteomics, which has the potential to deliver an enormously large source of molecular information, but has remained largely untapped today. We have a technology platform that broadly enables the scientific and the clinical community to access proteomic information with a combination of depth, unbiased, speed, and scale that we anticipate could create entire ecosystems with a potential impact that may be even larger than any other omics as we see today. Now, to kind of understand our ambitions to advance science and why we believe Seer's is a massive idea, it's important to understand the big gap in our current understanding of the proteome. So if you would please imagine with me that going from the genome toward the proteome, biology goes through a number of complex steps where you start off with roughly 20,000 genes, but you end up with about 1 million proteins. Now, this is in part because, as genes get transcribed into RNA and then later proteins are made, you make many different RNAs from a single gene, and even proteins themselves get modified after they're made. The end result is that you create an enormously complex number of proteins, and proteins themselves almost work if you would, in modules, in that they come together to form complexes and then they exert their biological effect. All of this is enormously complex. And it's been difficult. I mean, the fundamental challenge in the field has been that the existing proteomic technologies just not able to reach the depth, the breadth, and the scale to match this biological complexity. And in some ways, frankly, if you kind of just go back in time, just 15 years ago, this is very similar to what we went through with genomics. In other words, 15 years ago, before the advent of next generation sequencing, we were sort of where we are with proteomics today. Now we have a technology platform and a suite of products that we call Proteograph that we think is going to fundamentally change accessibility of the proteome in a similar way that the NGS did for the genome. And we think that the ability to access the proteome in an unbiased, deep, and rapid way, and at scale to do population scale studies, will create a vast new set of insights, methods, and end uses that, in time may have the potential to exceed the, the transformation and value creation that we actually saw in the genomic space. So we're thrilled to be considered for this category, but let me tell you why this award category is important for the industry. Now, we have made tremendous progress in understanding human health and treating human disease. And by the way, frankly, we have the genomics to thank for a lot of that. And at the same time, what we've achieved is just the tip of the iceberg to what is possible. There's so many diseases, so many patients still in dire need of safe and effective treatments. So much more progress that needs to be had. Now, smaller iterative advancements are how we do science, and they're important to continue progress. But big ideas are the only way to really move the needle and to bring about broad scale change and impact. So I think an award category that encourages and frankly celebrates big ideas is vitally important to our industry. Now, we think that what makes Seer stand out is the far-reaching implication of our technology and the platform and the product that we want to introduce to the market. The world has been searching really for an approach to access the proteome the way we've now able to access the genome. And this has been, super, super difficult to do. And we believe that our technology would enable, novel insights, methods that may even lead to creation of new companies and new ecosystem analogous to what the genome did. And so we're excited about the potential of Seer and humbled that we're being considered for this category.
Thanks a lot for having us on. I mean, it's definitely an honor to be selected as one of the finalists for the big idea. And I think for us with Squeeze, what we really feel is a big idea here is that it could truly unleash the power of cell therapies. So for us, this all started with an invention in the lab. When I was a PhD student at MIT working for Klaus Jensen and Bob Langer, we were working on various methods to try to deliver materials into cells. And what we discovered a bit by serendipity is that basically if you take cells and run them through a constriction at high speeds, you can disrupt the membrane temporarily. And to give you a sense for kind of how this works, if you were to shrink ourselves down to the size of a cell, it's akin to a sumo wrestler going through a hula hoop at the speed of sound. So it's a very quick event for a relatively large cell going through a tight constriction. But what this does is that it allows almost anything that's outside to be delivered inside the cell. Now, why this matters is that if you take a step back and think about cell therapies today, I would say they're very far from what they could be. So cells as a therapeutic could be by far more powerful than any biologic or small molecule ever could be. But as it stands, cell therapies are only really applicable to a small niche of disease areas where they work. They're very time consuming and complicated to administer, so it takes weeks or months to even produce them. And they're actually not the most pleasant thing to go through as a patient. So there's a lot of toxicity concerns involved, there's hospitalization. So really a far cry from what the potential of this next generation of therapeutics could be. With Squeeze, we solved the fundamental problem of just how do you engineer a cell effectively. And that really opens up a lot of biologies that are inaccessible right now and lets you implement it all in a practical way. So it takes cell therapies from what they are today to potentially having cell therapies where with Squeeze cell therapies, already what we do in the clinic is manufactured in under 24 hours. We can go after biologies that people can't, which lets you go after exponentially more patients relative to what's feasible today. And with what we've implemented, we actually don't require hospitalization. And from a side effect perspective, we expect it to be far more tolerable than what people have done so far. So really transforming what it means to have a cell therapy and actually realizing the potential of what these cell therapies can be as a new generation of medicine. So to give you a more detailed sense in what our first program is, so we have a partnership with Roche, which is an over $1 billion collaboration we've been working together on for about 5 years, which is around generating cancer vaccines. So if you think about the oncology world right now, the effects of the immune system is really pronounced, where if you can generate a patient's own immune response against a tumor, it can be extremely powerful. And that's the underpinning of a lot of these IO drugs that have made an impact for people right now. Now, a holy grail in the field has been cancer vaccination, which hasn't worked out. And what that means is having a system that can turn on your own immune response against the tumor in a specific way so that it only targets the tumor, doesn't turn on everything, which drives side effects. This is something that many have tried, and so far it hasn't really worked out in this field. But with Squeeze, we think we can solve a lot of the underlying biological problems that have prevented people from creating these effective cancer vaccines in the past. And already with what we've done in preclinical experiments is that we've shown this mechanism that Squeeze enables is about 1,000 times more powerful at inducing an immune response than what people have attempted so far. And that's what's in trial for us right now, which we would hope to see data towards the end of this year or early next on truly could this be transformative relative to what patients have done, what patients have had available so far. And if this works, it could be applicable to many, many patients within cancer and would be what we feel would be the start of these Squeeze cell therapies having an impact across many other patients in different indications down the line. Cell therapies are truly the next generation of medicines, and I don't think there's been as big of a shift in medicine since the advent of biologics in the 70s and the 80s. And I think what Squeeze really brings to the table as being the big idea that enables that transformation is that you can actually finally engineer anything you want about a cell and do it all in a practical format. So you can finally make dreams like cancer vaccines come true and do it all in a format which is actually very accessible to the patients. And so I think the big idea here is that you could truly make cell therapies practical and address underlying disease drivers such that they can actually transform the lives of potentially millions of people versus the very limited applications they have today. And I think cells as a new generation of therapeutics are a big thing for the world if we can get it to work.
Well, congratulations to all the finalists for this well-deserved recognition. And thanks to all of you for joining us today for this Meet the Finalists webcast. If you're hearing my voice, it means you're automatically registered and have access to the awards networking app. So I encourage you to take full advantage of it, connect and set up meetings with friends, colleagues, and business partners. We hope you enjoyed today's program and that you're registered for all the upcoming Meet the Finalists presentations. If so, we look forward to seeing you there. If not, there's still time. Just visit xconomy.com/national-awards. Either way, make sure to secure your complimentary registration to the 2020 Xconomy National Awards ceremony on September 24th. Let's all get together, have fun. It's going to be a lively evening as we announce this year's award winners. We'll see you there.